Tiny predators, huge promise. When antibiotics fail, viruses that eat bacteria fight back. The tiniest predators that heal, they called them living bullets. Tiny predators that eat bacteria. A century ago, they were a medical miracle that fizzled into obscurity. Today, as antibiotics fail, these microscopic hunters are quietly re-emerging, redesigned by genomics, delivered with precision, and sometimes carried in coolers across borders to save a single life. This is the story of bacteriophages, predators of microbes, and how a nearly lost therapy is staging a comeback that feels like science fiction made real. For most of the 20th century, antibiotics were our heroes. Penicillin, streptomycin, tetracycline, names that sounded like salvation. But microbes evolve fast. Decades of use, misuse, and global spread created superbugs that shrug off our best drugs. Worldwide, common infections have become harder to treat, and for some patients, antibiotics simply don't work anymore. Enter bacteriophages, or phages for short, viruses that specifically infect bacteria. They were discovered before antibiotics, briefly used in clinics, then sidelined. Now, armed with DNA sequencing and synthetic biology, researchers are bringing phage therapy back, more targeted, faster to design, and sometimes the only thing standing between a patient and a fatal infection. The story begins in the early 20th century. Scientists observed clear spots on bacterial cultures, areas where something invisible had killed the bacteria. Those clearings were caused by phages. Early clinicians, desperate for options, used phage preparations to treat infections with mixed success. Some patients improved dramatically, others did not. The variability and patchy standardization, combined with the arrival of reliable antibiotics, pushed phages to the margins. In the West, phage therapy waned. In parts of Eastern Europe and the former Soviet Union, however, phage institutes continued, accumulating decades of practical use and case reports that the Western world largely forgot. Fast forward to the present. Two technological revolutions have changed the game: rapid sequencing and synthetic biology. When a patient arrives with a drug-resistant infection, clinicians can now sequence the bacterial isolate and identify precisely which phage will attack it. Labs can screen collections of phages, or even engineer new ones, to match the bacterial strain. Phages are specific. They target particular bacterial species or even strains, which is both their superpower and a logistical challenge. Unlike broad-spectrum antibiotics that wipe out many bacteria, a well-chosen phage leaves the rest of the microbiome intact. That specificity reduces collateral damage, but requires a more bespoke approach. One of the most compelling modern stories involves a patient with a bone infection that resisted every antibiotic. Surgeons tried everything. As a last resort, doctors assembled a phage cocktail, tailored to the patient's bacteria. The treatment arrived in a cooler, was administered directly to the infected site, and within days the infection started to retreat. Over weeks, the patient recovered where antibiotics had failed. These rescue stories are dramatic, but they also raise tough questions. Which cases justify experimental use? How do you prove efficacy when each therapy is unique to a patient? Regulatory agencies are developing pathways for compassionate use, emergency applications, and small clinical trials to answer those questions. Phages are elegant in their simplicity. They attach to a bacterium, inject their genetic material, and hijack the host's machinery to make more phages. Then they burst out, killing the bacterium and releasing a new wave of predators. Some phages kill quickly. Others enter a temperate cycle, integrating into the bacterial genome and lying dormant. Therapeutic phages are typically the lytic kind, efficient killers. Researchers also study phage enzymes and bacterial receptors to predict resistance. Bacteria can evolve phage resistance, but scientists can respond by mixing phages, rotating them, or engineering their genomes. Phage therapy isn't magic. It's an arms race, but one where we now have tools to respond in near real time. Today's adaptations are where the story gets cinematic. Phage banks, curated libraries of sequenced phages, act like spare parts inventories. Hospitals and startup companies maintain panels of phages that can be screened quickly. There are engineered phages with altered host ranges, phages conjugated with antibiotics for synergy, and even phage-derived enzymes used as antimicrobial agents. Delivery matters. Topical phage gels for wounds, inhaled aerosols for lung infections, and intravenous or direct injection for deep-seated infections. Some teams are developing freeze-dried phage pills for oral administration, while others are exploring encapsulation to survive stomach acid. Logistics, cold chains, and production standards are modern hurdles that echo old vaccine and biologics supply challenges. Phage therapy raises policy knots. Because each phage treatment can be personalized, traditional randomized controlled trials are harder to run. Regulators are adapting with frameworks for compassionate use and adaptive trials. There's also the question of access. Advanced phage therapy is resource-intensive, and wealthier patients or research hospitals may get treatments first. Ethical frameworks must balance rapid access for desperate patients with the need for systematic evidence. International cooperation matters. Phages are everywhere, and many productive collaborations involve sharing phage libraries and expertise across borders. In some parts of the world, phage therapy is not experimental. It's an established option. Clinics in Eastern Europe and parts of Asia have used phage products for decades. Modern deployments, however, are scaling. Mobile labs, regional phage centers, and even commercial manufacturers are entering the field. Manufacturing phages to pharmaceutical standards requires clean facilities, standard assays, and rigorous testing for safety, purity, and absence of unwanted genes. Scaling production while keeping treatments tailored is a manufacturing puzzle. Standardized pipelines for discovery and rapid GMP production are emerging as the industry's next frontiers. No therapy is risk-free. Phages can trigger immune responses. High systemic doses may be neutralized by antibodies. There's the theoretical concern that phages could carry harmful genes between bacteria, a process called transduction. So careful screening for undesirable genes is mandatory. Bacteria will evolve resistance, which means clinicians must monitor and adapt. Another ecological question is whether releasing engineered phages into the environment could have unintended consequences. The answer isn't simple. Careful design, monitoring, and conservative deployment strategies can mitigate many risks, but vigilance and humility are required. At the heart of this technology are patients and families who face infections that take everything from them: mobility, time with loved ones, sometimes life itself. Their stories drive scientists to take risks, to build phage libraries, and to navigate regulation. Interviews with clinicians, even brief quotes, reveal the emotional gravity, the quiet desperation before a last-ditch phage treatment, and the relief when recovery begins. These human moments are why the scientific details, sequencing, cocktails, delivery, matter. They are not abstract curiosities. They are lifelines. Looking back, phage therapy is a lesson in scientific ebb and flow. What once seemed obsolete is now reborn through new tools and fresh urgency. The future will likely be hybrid. Antibiotics and phages used together, precision diagnostics guiding bespoke therapies, and international networks sharing phage resources. There will be triumphs and setbacks, trials and debates. But one thing is clear. When human ingenuity meets microbial diversity, new options emerge. Bacteriophages may not be a universal cure, but they are a vital new arrow in medicine's quiver. If you want a quick takeaway, antibiotics remain essential. Use them responsibly. Phage therapy is promising but still developing, and when it's the right tool, it can be life-saving. For deeper dives, check the episode notes for resources, patient stories, and links to phage centers. If you enjoyed this episode of Medical Mysteries Then and Now, subscribe, hit the bell, and leave a comment about which modern comeback you want us to investigate next.